Julia, whenever you're ready, you're welcome to begin.
All right. Hello. Thank you, Chrissy, for the wonderful introduction. I want to introduce myself. I am assuming you've probably met Dr. Jessica Budke. She's my advisor here at the University of Tennessee. I'm doing my master's on Fissidens mosses, which maybe you've seen some today while you've been working through specimens. Maybe you haven't. But today my talk will be on aquatic transitions and interspecific relationships in Fissidens mosses. Let's see. Oh, there we go. All right, so just as a little bit of an introduction to Fissidens, they are a high moisture loving moss that can be found anywhere from swamps to marshes and rivers and lakes. They can range from being fully aquatic to fully terrestrial. And I am particularly interested in these species that are fully aquatic, just kind of looking at their evolutionary history, trying to get kind of a better picture of their evolutionary history. So they can be found worldwide. It's estimated that there are 440 to 450 species that make up the group, and they are highly distinctive. Once you know what you're looking for, they're super easy to identify to genus in the field. So they have this unique disticus leaf arrangement that makes them appear flattened, as opposed to kind of the rosette shape and organization of leaves around the stem in most mosses. They also have a unique structure called the vaginate lamina, and I don't know if you can see my pointer or not, but it gives them their nickname, the pocket mosses. So it kind of makes them appear like they have a pocket. So an interesting phenomenon that can be seen in aquatic taxa that I investigate is morphological reduction. It's not an uncommon thing in mosses and can be seen in terrestrial species as well. But aquatic Fissidens have lost a number of characteristics, such as support structures to gain more flexibility in flowing waters. Morphological reduction is common in mosses. One of the examples of morphological reduction outside of aquatic habitats is when mosses move up trees, they tend to lose spore dispersal features. So their seta will be a lot shorter. You can kind of imagine they're up there in the tree. They don't need to kind of get the height that most mosses on the ground would need to disperse their spores. So aquatic taxa are morphologically reduced, as I mentioned before. They have that loss of support structures. So a little bit into the species that I'm focusing on. There's two subgroups of aquatic taxa, and historically they've been grouped based on those reduced features that I was just talking about instead of synapomorphies. So they've been grouped based on these lacking features. So they fall within two groups, the first group being subgenus Octodoceras, and the second group being section Sarawackia. So these aquatic species, again, have these morphological reductions, making it really difficult to tell them apart. The difference, for example, between Fissidens fontanus and Fissidens halianus is just a couple of cells from the costa, which is like the midrib of the leaf, to the apex of the leaf. So very, very small differences. So these two, again, are the two groups that all of the species within them are fully aquatic. So as the relationships between these two fully aquatic groups of Fissidens is convoluted, they've been my main focus, particularly because they're unique in this aquatic nature. So my main question in my thesis project has been whether or not members of these two groups are monophyletic in a phylogenetic tree. So to go about answering this question, I first had to determine what specimens to use. I did this using publicly available databases that you might be familiar with, like BioPortal, GBIF, and Tropicos. I have to thank all of the herbaria that lent me specimens. So I created kind of a list of a bunch of specimens that I wanted to request. I requested herbarium loan specimens from this list of herbaria that I have below. Got permission to destructively sample, because you can't just, request a specimen and not tell them that you're going to be destroying the sample for DNA extraction. And then once I got the loans, lost my notes. So once I got the loans, I verified the identity of the specimens underneath a microscope, annotated them, and then collected tissue for DNA extraction. So for these samples, I extracted all of the DNA, and then about 45 samples were sent off to a company called Rapid Genomics in Gainesville, Florida, to be sequenced. And at Rapid Genomics, they went through this kind of pipeline of DNA library prep, capture sequence enrichment, sequencing, and then they sent us those raw reads back. Rapid Genomics used a probe set called GoFlag 408 that is specifically for flagellated plants. So for those of you who aren't super familiar with mosses, they have flagellated sperm, which is really cool. Another group of plants that has that feature is ferns. So Rapid Genomics sequenced both the exon and the flanking regions of the DNA. And we got back raw reads from a specimen from 1842, which is pretty amazing and just points to the robustness of this probe set. So once we got all of these raw reads back, we put the reads through a pipeline of sorts based off of a publication that was published in 2020. We got back the raw reads, and there was about an average of 336 loci per sample. And during this pipeline, we removed samples that had less than 10 loci, just to clean up the data set. So after that, we lost about 3 samples, so that our updated sample size was 42. The average loci length was 758 base pairs, and the total length sequenced was 307,781 base pairs. So the next step was tree building. And some of you might be familiar with phylogenetic trees, some of you might not be. That's okay if you're not, it can be a little confusing, I'm very sorry. So the first tree that I created is a concatenated likelihood tree. So that's where I took multiple loci, aligned and concatenated them. I determined partitions, so like pieces and models for each of those pieces in the alignment. I inputted into this program called RaxML, and that output a maximum likelihood consensus tree. So I'll be showing you what that looks like. The second tree that I created is a coalescence-based tree. In this case, it was individual locus alignments that I input into RaxML again, but this created individual locus trees. And then we input all of the locus trees into another program called Astral, and that outputs a single species tree. And I actually, at the time that I made this presentation, I hadn't finished this tree yet, so I don't have it included in my presentation, but as of yesterday, I do have my coalescence-based Astral tree, so. All right, so let's take a look a little bit. This is what a phylogenetic tree looks like, or an example. This is going to be the maximum likelihood tree, so the first tree that I talked about. And everything is color-coded. There's a little key on the side you can see, but we're going to zoom in a little bit. And just to point out the colors that you should be looking at, as I mentioned, one of the subgroups that I'm looking at is subgenus Octodoceras, so that's going to be in the light blue. And then in the orange is going to be section Sarawackia, the other aquatic group that I mentioned. So if we zoom in a little bit and look at where subgenus Octodoceras came out in the light blue, oh, there we go. You can see most of it came out together except for this one species, Fissidens besuiensis. So a little bit odd. The second group, section Sarawackia, came out in 4 different spots. So back to my original question, whether or not members of subgenus Octodoceras and section Sarawackia were monophyletic. Oh, sorry. We can see that they don't come out monophyletic. Basuiensis, of course, is coming out a little bit separate from the rest of Octodoceras, and section Sarawackia is coming out in 3 separate spots here. So the broader implications of what I've done here and what I am continuing to do, as I'm not quite done with my project, is to contribute to the understanding of those habitat transitions in non-vascular plants, figure out the relationships between morphologically reduced species, and also determine systematic relationships between the species of Fissidens mosses. All right, and with that, I want to acknowledge all of the people who've helped me throughout the years. I have a wonderful undergraduate student, Julian Broughton, who helped me, as well as, of course, Margaret Oliver, the Go Flag Consortium, the UTK EEB department, Douglas Fuller, my committee members. I had funding from the Sharp Fund, and then, of course, all of the herbaria that allowed me to destructively sample their specimens. All right. And that is it. Are there any questions? I know that was a little bit overwhelming, so.
If anyone has any questions for Julia, you're welcome to either throw it into the chat or you can unmute yourself and ask. I just wanted to say thank you so much. It was an excellent presentation. Love learning more about mosses. But yes, we have a couple minutes left. So if anyone wants to throw out a question there, feel free to.
And you're also welcome to email me later if you'd like. I'm happy to answer any questions about what the process looks like for obtaining herbarium specimens, the process of my DNA sequencing. Yeah.
Julie, I'm not sure if you can see it in the chat, but there are a couple questions for you in there. Do you want me to read them aloud or do you want?
Oh, here we go. Okay,
sure.
I can see them now.
Okay. So the first one is from Mary, and it says, is there a proposed selective advantage to reduce leaves? And just apologize if you already answered it.
No, it's all good. Yeah, so there is a proposed advantage to the reduced leaves. We don't know 100%, but it is thought that the reduction in the leaves makes it a lot easier, especially the aquatic mosses a lot of times are found in flowing rivers. They can be found in lakes as well, but having that reduction helps the leaves flow better in the water and not just break. But that's still, there's still not 100% sure on it yet, so. And then, let's see, when did you know you wanted to study mosses? Ooh, can they disperse by fragments? That's a good question. Fragments, I'm not sure, but it is interesting. We hardly ever find the aquatic mosses with sporophytes on them. Not 100% sure why, but yeah. And then Elena asked when I knew I wanted to study mosses. That is a great question. Oh, here we go. Can you explain? So I'll answer the first question first. So I did my undergrad at Oberlin College in Ohio, and while I was there, I knew I wanted to go into botany. And my advisor told me I needed to pick a plant. And I told him I wanted to study sunflowers because I love sunflowers, and he said it was boring. So I randomly chose mosses, and here I am, and I love it. So, and another question: can you explain again the different relationship between Octodoceras and Sarawackia? Yeah. So, just do you want like the morphological differences or what kind of differences? because they come out separately in the tree, so they're two separate groups. Yes, he does. My advisor in undergrad said Asteraceae was boring, so that is why I am studying mosses, I guess. And I'm also happy to answer questions in the chat if everybody needs to, if I've taken up too much time. So just let me know.
No, we can go ahead and answer this last question. Timothy said morphological differences is
Oh,
what he
okay.
was mostly looking for.
Yeah, morphologically, so I explained it a little bit before, that there's a lot of morphological reduction within the mosses. And so just looking at the mosses in the field in a river, for example, it would be very difficult to tell the difference between Sarawackia and Octodoceras. They have different, if you slice the leaves very, very thin into very thin fragments, you can see that they have different cell arrangement in the costa. So that's the main difference, is there's an Octodoceras type costa. And then I don't remember the exact costa for Sarawackia, but Octodoceras has a very distinct arrangement of cells in the costa. So that's the that's the difference. You have to make one cell width thick cross sections of the leaves. So yeah.
Awesome. Well, Julia, thank you so much. We really appreciate your presentation, and I think we all can say thank you and really appreciate the time that you spent here with us today. Thank you so much.
Thank you.
All right, so we are going to still have another 45 minutes. I'm sorry, Matt's staring at me right now, guys. I'm getting like the heebie-jeebies. We're going to continue transcribing, so you're welcome to go back into your breakout room, work on transcription for about 40 more minutes. If you have any questions, feel free to drop them in the chats or unmute. And if you find any interesting specimens or collectors or localities where the collections are from, shout them out to the group too, make it more interesting. And I'll see you guys again in about 40 minutes, okay? Feel free to move to the other breakout room. You don't need an invitation. If you go to breakout rooms, you can just select to join. Have fun, guys.